Okay, we are going to continue talking about cellular division. We were discussing mitosis and the process of mitosis. Mitosis is the way our somatic cells divide. Our somatic cells, meaning our body cells, all of the cells in our body, divide through a process known as mitosis, creating clones, creating identical daughter cells. These identical daughter cells are every cell in our body has the exact same DNA, the exact same chemical makeup. It's just that DNA that's turned on and turned off in those cells is what makes all of our cells different, from our muscle cells to nervous cells to blood or bone or anything like that. Well, what we're going to talk about today, though, is meiosis. Meiosis is the cellular division that occurs inside of our gametes, inside of our sex cells. Our eggs and our sperm cells are produced through this process known as meiosis. Through meiosis, we create we create 4 daughter cells. And those 4 daughter cells are all unique in their DNA makeup. They are all genetically unique. They do not have identical DNA in those chromosomes. When we discuss meiosis, there are a couple points that we need to cover beforehand. Through genetics, we inherit certain characteristics. We inherit certain properties from our mother and from our father. Just like we were talking about how you get a left and a right foot, you have 2 different types of chromosomes in your cells. 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s. And during meiosis, these chromosomes, for example, they're paired up during the process of meiosis, and then they divide off to where you end up with only one cell that has only one of each chromosome, this being that daughter cell. We are cutting that chromosomal number in half, creating a haploid daughter cell. Okay? This would be diploid, where there are 2 of every chromosome, and this would be haploid, where there is 1 of every chromosome. And this is our objective, to cut that chromosomal number in half to create these haploid daughter cells. In organisms that reproduce sexually, meiosis occurs. It only occurs in sexually reproducing organisms, where there has to be the cutting in half of the chromosomal number so that through sexual fertilization, that chromosome number is restored. That diploid number of chromosomes is restored. Bacterial cells, they go through mitosis. They go through asexual reproduction. All of the cells in your body is going through mitosis. That's asexual reproduction. But when a sperm and an egg come together, that's that sexual reproduction, that joining together 2 cells to create this diploid number again. And so as we go through meiosis, we're going to look at this breaking in half, or creating only half a number of chromosomes in those daughter cells. That's our objective. Well, cells that go through meiosis still go through the cell cycle. They start off in interphase. There are, for females, our egg cells develop while we are still in utero, in the uterus of our mother. Okay, about 16 weeks into gestation, when a mother is only 4 months pregnant, if she's pregnant with a daughter, her daughter has all of the egg cells that child will ever have in her entire life. So you are born with all of your egg cells, and it's not until you reach puberty that you start releasing those egg cells. And then for 30 years, we release the egg cells, and then we go into a process known as menopause, where the hormones are not there to allow those egg cells to be released anymore. So by the time we're 45, we've got 45-year-old eggs. By the time we're 60, we're no longer releasing those eggs, but we do still have some. We do still have eggs. And so for a woman to be 65 years old and to have a baby, it's not an alien baby like, the tabloids want to say. It's probably she harvested an egg, went through, in vitro fertilization and everything else, even though she wants the papers to think, oh no, it was natural fertilization. Probably was not natural fertilization. It probably was in vitro or something like that, where she's, they harvested the eggs, fertilized them, and then reimplanted them. But we do still have those eggs, even if they aren't being released hormonally. On the other hand, males don't start developing their sperm cells until they do reach puberty. And at puberty, when they start producing their sperm cells, they will produce them until they die. It doesn't matter. Age does not matter. They are continuously, constantly, forever going through meiosis. They are constantly producing new sperm cells. And in realistic terms, there's about, if you have less than 8 million sperm cells in a milliliter sample, you're considered infertile. So what that means is basically there are millions upon millions upon millions of sperm cells being produced daily in every male, constantly being produced. And it's because of this process known as meiosis. So those stem cells, those cells that are going through, getting ready to produce our egg cells, getting ready to produce our sperm cells, are still going through mitosis. They're still duplicating themselves. They're still going through mitosis, especially in the males. In the females, it's just in utero, but in the males, it's happening all the time. You're constantly still going through mitosis of these original diploid cells, these cells that we actually call spermatogonium. Okay? They're constantly going through mitosis. And then every once in a while, those cells will then diverge off and start heading into the meiosis process to become mature sperm cells. So that's what we're going to look at. That's how we're going to discuss this process of meiosis. Well, if a cell is still going through the cell cycle, then it's still going through G1, it's still going through S, and it's still going through G2, getting ready to enter into the process of meiosis. So if it's going through G1, S, and G2, these chromosomes here are going to do what? What are they going to do? They're going to duplicate. So they're going to end up with, a copy of every one of these chromosomes here, held together by those centromeres. Okay? Well, looking at this, you can see that I now have the 4 chromatids, right? Or I have a pair of the same chromosomes. Well, this pair of chromosomes, the 2 number 1 chromosomes, the 2 number 2 chromosomes, the 2 number 3 chromosomes, and so on and so on and so on, these are known as homologous chromosomes. Homo means the same. Homologous chromosomes. These are the same chromosomes. So we have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 21s, 2 number 22s. Okay? We have 23 pairs of homologous chromosomes. We have 23 pairs of homologous chromosomes. Meaning we have 23 different types of chromosomes. Out of those 23 pairs, chromosomes number 1 through 22, these are actually known as autosomes. Chromosomes number 1 through 22 are considered autosomes, meaning they belong to self. They're self chromosomes. These are pretty much the same in every human being. We have 22 pairs of autosomes. They might have slight variations. One might say blonde hair, one might say red hair, one might say I can roll my tongue, the other one says I have detached earlobes. And there's variations of these autosomes, but they're basically still giving us the same information for the human being. Chromosome number 23, however, is known as our sex chromosomes. Chromosome number 23 has homologous pairs of sex chromosomes. Now, there's a slight variation in our sex chromosomes. Females have 2 of the same chromosomes. We have 2 X chromosomes, which means, they're basically very long chromosomes that, form a homologous pair. They complement each other. Think of homologous pairs being complements of each other, or whatever. So a female actually has 2 X chromosomes, 2 X sex chromosomes. A male, on the other hand, has the X and the Y chromosome. So these are not the same. These are not actually considered homologous chromosomes like the rest of them are, because the X chromosome, yes, is nice and very long, but the Y chromosome actually is a very small chromosome. So they do not pair up exactly the same, but they are the 2 homologous chromosomes for our sex chromosomes. So that's kind of what makes them pair up. And realistically, what allows these chromosomes to say, well, this is chromosome 1 and this is chromosome 1, what allows these 2 to pair up is that there's special marks or genes on them that correspond with each other. If there's a gene here and a gene here and here and here and here, well, it's the same thing on this one. And so they're going to match up that way. They match up according to their genes. Well, if this X chromosome has genes like this all the way down it, the Y chromosome actually has complementary matches on a certain part of its chromosome that allows it to pair up. Does that make sense? Okay. So naturally, in our bodies, during meiosis, our homologous chromosomes we're going to see actually come together and form these pairs. Now, scientists do it in general all on their own, too. It's known as a karyotyping. A karyotype is whenever, they're interested in looking at maybe some kind of genetic disorder, or if you have a history of, for example, cystic fibrosis in your family or something like that, which is a genetic disorder. It's something that, can be very serious if you have a child with cystic fibrosis. So what they can do is they can look at a karyotype. A karyotype is taking a sample of your blood, pulling out of your white blood cells your chromosomes, and looking at them and actually pairing them up according to their homologous pairs. And when we do it manually, when a scientist is doing it, they actually have to find, they separate them out almost according to the length of the chromosome to start with. And then you can see the different colorings on here. Those are the complementary genes. Those are saying, well, this is for hair, and this is for tongue, and this is for ear, and this is for eye, or whatever, and they match them up. Now, we have over 300,000 different genes on our chromosomes. We have a tremendous amount of genes on our chromosomes. But when we pair them up, you can see also they pair up pretty much according to length. Number 21 down here is a very small chromosome compared to number 1. And so that's kind of an easy way of pairing them up, too, for a scientist. They pair them up according to size, and then they start looking at those genes, making sure that they're the same genes. Because 1 can kind of be confused for 2, couldn't it, when we're looking at length? But this is a karyotype. They will do a karyotype when they're looking for any kind of genetic disorder, or whenever there is somebody that's pregnant. Every woman in the state of Florida, if they're over the age of 35, is pretty much ordered to have an amniocentesis now. Over the age of 35. Whether you're 35 when you're pregnant or 35 if you're going to be 35 when you deliver. You might be turning 35 the day before you're supposed to be delivered. Well, you have to have an amniocentesis done now. What does that mean? They're actually going to draw out some of the amniotic fluid inside of the uterus to do a genetic test on, to see if there by chance happens to be 3 number 21 chromosomes, which would equate out to Down syndrome, or if there's any other genetic abnormalities in this karyotype. If you're having difficulties conceiving, they might do a karyotype. I know in my own personal history, I had 3 miscarriages before I became pregnant with my daughter. 3 miscarriages are considered excessive. 2, it might be normal. 3 becomes excessive. They thought, well, maybe there was something wrong with my chromosomes. So they did a karyotype on me and my husband. Then they were going to start looking at my uterus saying, okay, well, I was up to about 9, 10 weeks before I miscarried every time. They were going to start to see, is there a reason why it's not implanting solidly or things like that. Those are tests that they run. They see, why is this happening? Why is this occurring? Is there something abnormal in the genetics, or is there something else going on? Needless to say, there wasn't anything going on after third time, because 3 miscarriages, 3 babies. Could you imagine if I had 6 kids? Thank you. 3 is enough. But that's another reason why you would do a test like this. If you wanted to know if you happen to be a carrier for BRCA1 gene, the breast cancer gene, they'd do a karyotype. They'd look at it. Breast cancer occurs on several different chromosomes. They'd look at it, see what's going on there. That's the idea behind it. Okay? Any questions on that so far? Okay. Well, what allows them to pair up? What allows our chromosomes to pair up basically is the arrangement of those genes. The genes are found at a very specific location on our chromosome. This location is known as the locus. Okay? Kind of makes sense. Locus for location. So when we talk about where is a gene located, we say, where is the locus? Where is its loci? What chromosome is it found on? What position is it found on? And if you ever have the desire to look at chromosomes, there's a great website from NIH dealing with the Human Genome Project, looking at every chromosome. You can find any gene on any chromosome that you have of interest. If you're looking for that obese gene, if you're looking for that Down syndrome gene, if you're looking for, I guess it's not a Down syndrome gene, cystic fibrosis gene or breast cancer gene, or you can go to the Human Genome Project's website, punch in chromosome 15, and they'd give you every little bit of information you ever wanted to know about chromosome 15. Or you can type in Alzheimer, and they would give you every chromosome that there is a gene associated with Alzheimer on, and what causes it, and if it's a mutated protein or if it's a normally occurring protein that's no longer functioning or something like that. Okay, so you can go to these websites and find all kinds of incredible information. You can go to a website known as DNA Interactive. DNA Interactive also will give you information on these genes and these chromosomes. Gene splitting does happen. It depends on what variation you're if it does happen naturally or it could happen man-made wise. It goes along with gene therapy, things like that. Yes, it is. Very difficult. Very difficult. It's something that they would do more in utero than they would afterwards. So, okay. Try and catch everything before as best they can. Yep. That's their objective. Okay. Any other questions on these chromosomes right now? Locus is basically the location of the gene on the chromosome. We have over 300,000 genes. Each gene is going to produce a protein. Basically, proteins are everything that are expressed. So where those genes are found is known as the locus on the chromosome. Okay. missing. So what we're going to do is we're going to talk about how this, these two haploid cells here are going to be formed. Here we have our testes and the ovaries. Inside we find our egg cells and we find our sperm cells. They are haploid cells. The sperm, carrying 23 chromosomes, will fertilize the egg, carrying 23 chromosomes, and restore that diploid number of chromosomes, the 46 total chromosomes, or the 23 pairs. Imagine your mom and dad, the day they were married, were both given a set of encyclopedias. Okay? Dad got World Book Encyclopedia, Mom got Encyclopedia Britannica. That incredible wedding gift, right? 2 sets of encyclopedias. Well, whenever they had you, they kind of mixed their volumes together. Okay? They took their 23 volumes, and each one of them had 23 volumes, put them together, and gave them to you. Okay? So now you are this combination of World Book Encyclopedia and Encyclopedia Britannica. Okay? Now, if you opened up both of those books to the same topic, and you start reading about that, that same topic, and I know you guys probably don't even know what encyclopedias are anymore because of the internet and everything else. Who needs an encyclopedia, right? But they were books that carried knowledge at one point. Huh? They're expensive too, yeah. So you open up this book and you want to find out all about, I don't know, give me a topic. What do you want to learn about? Huh? Meiosis. No. Huh? George Washington, okay? So you open up the World Book Encyclopedia and you're saying, George Washington, the father of our country, he was the first president, led us through the Revolutionary War and made this country what it is today. And then you open up the other book, the Britannica Encyclopedia. It says George Washington. He's on the $1 bill. Okay. He was the first president, but he didn't run for a third term because he knew when to go out, on top versus extending his stay. And he got us through a pretty important war known as the Revolutionary War, right? So you're kind of getting the same information, right? It might not be exactly the same, but it's pretty darn close, isn't it? And you could probably write a pretty decent report on George Washington using the information from both of these books. Now, granted, there's probably a little bit more than that on them, but you get where I'm coming from. Our DNA, even though we've got one coming from mom and one coming from dad, it's still giving us pretty much the same information. Facts may change. Facts are going to be the same, but the way they're presented are going to be changed just a little bit. And that's what our chromosomes are all about. And that's what inheritance is all about. We look kind of like mom, kind of like dad, We got a little bit of both of them being expressed. Maybe more dominantly mom or more dominantly dad, but we've got that little bit of expression going on. We're kind of pulling them both together. And that's what's coming off of these genes here. And that's what's happening in this diploid cell. Now, when you go to get married, you take those combination of books that you've got, and you're going to only be allowed to take 23 of them with you into your new marriage. So you've got to kind of pick and choose. You get to pick out Volume A of Britannica and Volume B of World Book or something like that. But you can only take 23 of them with you into your new marriage. See where I'm going? And that's what's going to be passed on to your next offspring. Does that make sense? So that's kind of what we're looking at here. So we've got this diploid cell. It's also known as a zygote whenever the egg and sperm fertilize, create this first diploid cell. It's the zygote. That's what's going to go through mitosis over and over and over and over and over again, building the organism. And inside the organism, just that one set of cell will go through meiosis. Will develop into meiosis. Okay? Questions on that? Okay, well, let's look at meiosis in a, just a quick overview type of appearance here. What's happening during the process of meiosis is in that diploid cell, in that original 46 chromosome cell, we will see that our chromosomes will pair up. Where'd my light go? Okay. These homologous chromosomes pair up. They come together. They form a molecule, not really. They pair up through what's known as synapsis. So if this is my chromosome 1 World Book, and this is my chromosome 1, Encyclopedia Britannica, they're going to come together, okay? And they go through what's known as a synapsis. And then through the process of meiosis, we're going to separate them out, the pairs, the homologous pairs will separate out, and then they're still duplicated, right? Because originally they're duplicated. We have 2 of every chromosome. Those duplicated chromosomes will then separate out. And now we're going to end up with 4 daughter cells, 4 completely different cells. Okay? So you can see we've got these homologous chromosomes pairing up. The homologs will separate, creating 2 daughter cells in this first cell division that has that duplicated chromosome, but now only 1 instead of 2. Okay? It has only 1 instead of 2. And then those duplicated chromosomes, the sister chromatids, will separate out and give us 4 daughter cells with unduplicated chromosomes. So that's our objective. Remember, meiosis has 2 cell divisions. The first cell division separates the homologous chromosomes, and the second cell division separates the duplicated chromosomes to where they're unduplicated. Kind of just like what we saw in mitosis, right? Mitosis separated those duplicated chromosomes into unduplicated chromosomes. Questions on that? Yes, ma'am.
Where on that chart would
Synapsis occurs up here when the 2 homologous chromosomes join together. Right here is where synapsis is. Okay? Now, they show this on the diagrams and in the charts that we're going to look at, the different chromosomes in different colors. Blue and red are the colors that they're using. Imagine the blue ones, say, for example, were the chromosomes. We're talking about your chromosomes, your sex cells, your eggs, your sperm, your cells. Okay? Say the blue ones were chromosomes that your father gave you. And the red ones were chromosomes that your mother gave you. That's that mixed, those books that they gave you. Okay? So your World Book and your Britannica. That's what's going on here. Now what we're going to do is look in detail about what's happening through these steps of meiosis. Meiosis, 2 cell divisions. The 2 cell divisions are basically known as meiosis 1 and meiosis 2. Okay, why do I feel like I'm spelling this wrong? I am. Meiosis 1 and meiosis 2. Okay? Now, each cell division is further subdivided. It's going to be challenging for you guys. Okay? Meiosis 1 is subdivided into prophase 1, metaphase 1. There's no prometaphase in meiosis because in actuality, if you ever went into like an embryology class down the road or something like that, prophase 1 is actually further subdivided into 5 steps. And so there's no prometaphase, in other words, in meiosis. So we have metaphase 1, anaphase 1, and telophase 1. What do you think happens at the end of telophase 1? Cytokinesis. The splitting of the cytoplasm. Now, here's a very important fact to remember. At the end of telophase 1, or end of meiosis 1 and cytokinesis, the cell does not It does not go back into interphase. It does not. The cell goes into like a very short, mild resting period known as interkinesis between divisions. Okay? So we go through this interkinesis before we enter into meiosis 2. So we can put here interkinesis. Okay? Kind of between meiosis 1 and meiosis 2 is this period known as interkinesis, between divisions. And now meiosis 2. I bet you're a smart enough group to figure out the steps of meiosis 2. What do you think? Prophase 2, metaphase 2, anaphase 2, and telophase 2. And then once again, cytokinesis. Okay? It does not go back into interphase. It goes into interkinesis instead, which is just kind of this in-between phase, in-between step. Now, the important thing to remember is we're talking meiosis. Meiosis is different than mitosis. When I say, when does this occur, you need to tell me, well, this is occurring during meiosis. Well, when during meiosis? Well, anaphase 2 of meiosis, or prophase 1 of meiosis, or metaphase of mitosis. Anytime you're talking about these processes or these steps, you need to distinguish each phase either by prophase, prophase 1, or prophase 2. Metaphase, metaphase 1, metaphase 2. Get where I'm going with that? Because every step, there's something different happening. Prophase of mitosis and prophase 1 of meiosis are very similar, and yet different things happen. The same thing as prophase and prophase 2. Prophase of mitosis and prophase of 2 of meiosis are identical, other than the fact that mitosis you have a diploid cell, and in meiosis it's a haploid cell. Okay? So we're going to see these differences. Well, let's start off looking at this. We have interphase. During interphase, we have G1, we have S, where our chromosomes are duplicating, and then we have our G2 phase. At the end of our G2 phase, if they say, okay, is this cell ready to go into meiosis? And if the answer is yes, if all those checks have been made, and we've done everything we need to do, then these centrosomes with the centrioles inside of them will duplicate, just like we saw in mitosis. And we will enter into prophase 1 of meiosis. Prophase 1 of meiosis. And that's what we've got here. Now, a lot of things are happening during prophase 1. The centrosomes with the centrioles are going to start to migrate to opposite poles. As they migrate, they're putting out these spindle fibers. These spindle fibers eventually will become kinetochores and non-kinetochores microtubules. Sound familiar? Another thing that happens during prophase 1 is our chromosomes condense down. They coil up. They get really tightly wound together. Sound familiar? A third thing that happens during prophase 1, what do you think? The nuclear envelope breaks down. The nuclear envelope starts to disappear, disintegrates. The nucleolus disappear. These chromosomes are now going to just be floating around basically in the cytosol. Okay? So, those same things happened in prophase of mitosis, didn't they? Well, here's where the differences come into play. During prophase 1, two more things actually happen. During prophase 1, the homologous chromosomes will pair up. They will find their partner. They will find their homologous chromosome. If I've got chromosome 1 and chromosome 1, they're going to pair up. They're going to come together, form this homologous pair. Synapse. This is where synapsis occurs. This is synapsis. And as you see this, how many chromatids do I have here? How many chromatids? I've got 4 chromatids. What I've got is a tetrad. Tetra is 4, right? Di, tri, tetra, right? Quadra, tetra, same thing. Tetrad is 4. I have formed a tetrad. These homologous chromosomes pair up and form a tetrad through this synapsis. So that's another thing that happens during prophase 1. But that's still not that incredible, is it? But here's the thing. During prophase 1, and only during prophase 1 of meiosis, after this synapse has occurred, after these tetrads have formed, a phenomenon happens that is only happening in sexually reproducing organisms in meiosis 1, in prophase 1. And what that is known as is crossing over. Crossing over. What is happening is that this genetic information, Encyclopedia Britannica and World Book Encyclopedia, come together. And now what you're going to do is you're going to rip out 20 pages out of one book and 20 pages out of the other book and mix them up. I'm going to take these 20 pages and put them here and these 20 pages and put them here. I'm going to cross them over. And that's exactly what happens with these chromosomes. We see this crossing over here. We form this little X. This X is known as the chiasmata, which represents crossing over. Crossing over is an exchange of genetic information between non-sister chromatids. An exchange of genetic information between non-sister chromatids, between the blue and the red chromosome. Non-sister chromatids. The reds, those are sister chromatids, right? The 2 blues, sister chromatids. 1 red, 1 blue, non-sister chromatids. Crossing over. An exchange of genetic information between 2 non-sister chromatids. And so now what happens is this blue chromosome actually ends up carrying a piece of red DNA, and this red chromosome carries this piece of blue DNA. And so now this is a completely different chromosome than this, isn't it? It is no longer identical. It is no longer the same chromosome. And if I put letters up here, if I have A, B, B, C, C, D, D, E, E. And while over here, I'm going to have the same things, but I'll do them in small letters. You see how I've now exchanged that information? This is a slight variation in my gene. This says, the small d, for example, says that I cannot roll my tongue. And the large D, the capital D, says, yes, I can. I can't roll my tongue, I can roll my tongue. Well, now, if my son ends up with this chromosome, he's not going to be able to roll his tongue, is he? Or if he ends up with this one, he still will. He can roll his tongue. Does it make a difference in life? No, but it's a genetic inheritance.